Hello, everyone. Can you hear me okay? I'd just like to thank everyone for giving me the opportunity to present our ongoing work using saturation mutagenesis to look at how pleiotropy works in a cancer setting. So pleiotropy very generally is when mutations in one protein can affect multiple different traits. And so this is very common in biology, and especially in cancer genes and a lot of our targets that we care about in cancer therapeutically. So for example, this cytosolic DNA sensor STING, its name comes from a stimulator for interferon genes. And so this interferon function is thought to be tumor suppressive. So if the cell has DNA damage that leaks into the cytosol, turns on interferon, the immune system kills those cells. However, we don't find that STING is mutated in advanced cancers like you might expect from a tumor suppressor. And instead, in these chromosomally unstable advanced tumor cells, STING is actually chronically activated. And in those settings, instead of having an interferon response, these cells have an NF-kappa B response and an ER stress response. And those responses are both very oncogenic, and they drive metastatic progression. So what we found with STING is how you activate it really matters for what kind of signal you get. So in blue, we take fibroblasts and we natively stimulate them with cGAP, the ligand. We get a very strong interferon response. But if you keep stimulating them over and over again, after 5 days, you switch to this red response, that interferon is gone, and now they switch to a very strong ER stress and NF-kappa B stress response. So there's this context dependency on how STING is actually turned on that changes what its function is. And we found in these advanced metastatic chromosomally unstable mouse tumor models that if you inhibit STING, these mice live a lot longer. So there's this ongoing debate whether we should activate STING or if we should inhibit STING in a cancer setting. Now, pleiotropy we could explain through kind of a classical domain structure of STING. So this is the structure of the homodimer of STING. You can see the transmembrane in green. There's a dimerization membrane in blue, and there's this ligand binding domain in brown here. And the C-terminal domain in red is typically what we think of as the interferon domain. So this is where the interferon function is coming from. But we don't really know where the ER stress response and the NF-kappa B response or the apoptosis or ELC, all these other different functions of STING, what they're related to in the structure. And there's a lot of debate going on about what residues specifically are controlling those functions. So this has led to these three main questions that we're aiming to answer here. One is, can we map which residues individually contribute to each of these different functions? And the second is, are these functions independent, right? So are there specific residues for specific functions, or are there some residues that are controlling multiple functions together? And lastly, we want to know, could we allosterically inhibit these specific functions, right? So if we could find a place on STING where you could bind a small molecule to that turned off those oncogenic functions but activated or maintained the tumor suppressive functions, this would really be the ideal drug for us rather than looking at agonists or antagonists specifically. So to approach this problem, we're using a saturation mutagenesis approach that we call SatSeq. And what we do is we directly synthesize all the different variants of STING, where we go one amino acid at a time. We mutate to all other 19 amino acids. So for STING, it's about 378 amino acids. That gives us 7.2 thousand different variants, a protein that we directly synthesize. And we associate that with a DNA barcode in the 3' UTR of those variants. And it's a double barcode, so the blue barcode on the right side here is a known sequence that's synthesized with the variant that tells us what amino acid is mutated. And that red barcode on the left is a random 16-enmer that acts like a UMI. So every clone, every cell has a unique overall barcode. And then we can use long-read sequencing to map that unique barcode back to the specific mutation in that variant. And we clone this into this modular system where we have a bunch of different promoters, so we can tune our expression level of our transgene into a lentivector. And what we've shown with 4T1 is if you knock out STING, you get a really great decrease in its ability to form metastatic lesions. But if you complement it back in with this system, you get a dose-dependent response, restoring that metastatic phenotype. So we can tweak how much of our transgene we put in. And all of these transgenes have a 10x capture sequence 1 embedded just downstream of the barcode between the UPRE element. so that we can capture this transgene in 10x single-cell RNA sequencing. So this gets us a full transcriptional profile of all our different 7,000 variants. And we can also quantify these barcodes with sequencing to look at changes in relative abundance. So we can take our library of different variants, inject it into a mouse, and then harvest the primary and metastatic tumors from that mouse and see how the relative abundance of each of the variants changes from pre-injection to the actual in vivo system to see which variants are selected for in those environments. Similarly, we can do the same sort of thing with immunofluorescence and FACS sorting. So you can take your library, stain it with an antibody, FACS sort it, and look at how what protein levels are changing. And this gives you a more mechanistic explanation for what those phenotypes are coming from, right? So this is what the data actually looks like. The variants are all here on the bottom, and you can see on the far left, there's some variants that always are lost. So this is a replicate of 10 different mice, and each of these dots is the change in relative abundance for that position in that mouse. And so some of the positions are always lost in all mice, so they're not able, if we mutate that position, they're not able to form metastatic lesions anymore. And you can see one of these ones highlighted here at the bottom, RRDD, and Kenny, maybe you find this interesting because this variant specifically disrupts the binding of STING with STIM1. So you lose that binding partner of the protein, and what happens is we can validate, if we just screen this mutant on its own, we do see that it loses its metastatic potential. And in the middle, we have a large number of positions that if you mutate them, there's a slightly negative or neutral effect. So this is kind of expected. And on the far right side, over here in red, there's some positions that if you mutate them, we always get an increase in metastatic potential, right? And we can use that transcriptional data that we have because we can do the single cell for all of these and extract that position, and we can see that in this case, those variants that are mutated there have an increase in NF-kappa B signaling, which is pro-oncogenic, but a decrease in their interferon response. And if we look at this transcriptional data a little closer, what we've done is we've treated our library with DMXAA, which is a STING agonist, to turn everything on. And we have a control where everything's treated in DMSO, so everything's kind of baseline. The DMSO is in red, and the DMXAA treatment is in blue. And so you can see this very big shift in transcription when we activate STING. But the replicates here are on the left, and they're all still fairly well mixed. So it's not a dosing-independent effect. We've kind of jury-rigged cell assign, so instead of calling cell types, we've had it call pathways. And so what you can see in those DMSO cells, there's no real distinct pathways turned on. So most of them are still unassigned. Sometimes they have an ER response, sometimes it's mostly autophagy, but there's no overall pathway. But when we treat it with DMXAA, you get this big shift to an interferon response. And if we look at the individual variant positions, how far they're shifting in response to DMXAA, we can plot that here as just a Euclidean gene space distance. And you can see that there's some variants here that don't have a big response at all on the far left, and there's some variants on the far right that have a very big response. I want to highlight a few specific ones. So in gray there's a stop control. So this is a variant that has 2 stop codons, so it doesn't produce any protein. And you can see it doesn't have a big response. So this isn't surprising, but it's a good control to have. And our reference sequence, which is wild type, it has a very robust stop, a very robust response to DMXAA, as we would expect. But there's a few here that have a very attenuated response. So I229A is actually in particularly interesting because DMXAA failed its clinical trials because that mutation is present in human STING, which renders DMXAA not useful in humans. So we see if we introduce the same mutation into mouse STING, you lose responsiveness to DMXAA. And conversely, this variant S161A, in humans, if you introduce that mutation, you return sensitivity to DMXAA even in the presence of the I229A mutation. And we find if you introduce that mutation into mouse STING, it becomes even more sensitive to DMXAA. So these are in particularly interesting for us as a transitional medicine because all of these hypoactivating mutations are the mutations that are you're going to find is resistance emerging when we actually treat with the drug. And these hyperactivating mutations are particularly interesting for us because those would be your ideal patients in clinical trials. If you could screen your patients and you identify people who have those mutations, they're great candidates for your drug. So in conclusion, I'd just like to say, SatSeq, this is a modular pathway. We built it so that we can kind of tweak and put it into different systems very easily without having to resynthesize the whole library. And we are able to detect specific mutations that confer an advantage or a disadvantage in an in vivo setting. And lastly, we're able to detect hyper and hyporesponsive mutants to specific drugs. So I'd like to thank you for your attention. And I'd like to specifically thank people in our lab, the Lockney lab, and the Bakum lab, in particular, Amelia Onstead and Mercedes.
I'll maybe start with one question. Is this functioning, so there's just a single variant expressed per cell?
Yes.
Would you get a different result if there were two different alleles?
If you had multiple different variants per cell, you start getting confounding effect and the signal starts to get muddled. So we do the same thing as the base editors and everyone else seems to do, is like try to limit the MOI so that you only get one copy per cell. But that's a good point. We specifically try to avoid that. You can occasionally see in the single cell data multiple barcodes per cell. And so those are those like rare less than 5% that are doubles. And we try to exclude them from the data so that we don't have those kind of problems.
Yeah, thank you very much for this very interesting talk. Two brief questions, if I may. In human patients, obviously the inter-patient heterogeneity is one of the biggest challenges in metastasis that we see. So you tested 10 different mice, if I understood your data correctly. Mm-hmm. How different are the results between the different mice? Because I think most of the other data were aggregated data that you have shown. So do you always find the same mutations doing the same thing in different mice, or do you see differences?
Does this go back? No, I guess not. Hey. So there's a large range of them where you get a lot of variability. So for us, the biggest problem, there it goes. Oh, okay, never mind. The biggest problem is that we have a large library size, and you can only inject so many cells per mouse. And so there's always the concern that there's a sampling bias. So we do 10 mice here. We're also doing 30 mice in downstream experiments. We try to keep that mouse level really high. There's only a certain number of variants where it's always the same in the mice. And those are really strong effects. So there's like a handful that are always positive, and there's more that are always negative. But for the most of them, you get a lot of variability, and we think it's usually sampling bias. So what we do is we do an in silico bootstrapping test with all our sequencing. So we do the bootstrapping to check that we have enough sequencing depth as well. But then in addition, we do an in silico experiment from the initial relative abundance where we subsample the same number of cells we inject from those reads, and we see what the expected distribution will look like just from the sampling bias. And then if the observed change in relative abundance is within that distribution, we just consider it null, because it could just be from sampling.
Did you have the chance to look at different metastatic sites?
So not in this model. These are specifically cell lines that are breast cancer to lung models. It's something that is really interesting, but we've had an issue with, we want to have a lot of tumor cells in the metastatic site to keep that, basically that subsampling not too strong as well. So when we've looked at really small micromets, this has been a problem. So at the moment, we're keeping it to where we have nice big lung mets. One more.
Just based off of like those really strong mutations you found and based off of the structure, predicted structure, do you have some insight about structurally, like what's happening to sensitize? Like, for example, are the really strong responders, are their basal levels of activity also higher? Or is it like a compound specific effect?
So far, we haven't found, like, so here you see the response distance mapped onto the 3D structure in this diagram here, which I don't really have time to talk about this kind of depth, but we don't see like a very strong distinct clustering of the hyper-responsive or hypo-responsive to specific 3D locations. So this was kind of the a priori assumption that we had made, was that where the drug was going to bind was going to be all hypo-responders. And it would be a very clear signal of where the drug sticks. But what we found is that there's a lot of mutations that seem to stabilize or destabilize the overall conformation of STING, which is like there's a very strong conformational change that occurs when it activates that is necessary for those downstream responses. And so those seem to stabilize or destabilize that. We've been doing a lot of in silico structural predictions with CoLabFold and AlphaFold2. And we see these kind of differences in stabilization of the different conformations, but we're not sure how much of that is like artifacts from the machine learning or not. So I haven't wanted to talk about that quite yet, but hopefully that's gonna, we can validate some of those soon, is our goal.
Very cool, thanks.
All right, well, thanks very much. Great talk, Matthew.